In the autumn of 1937, at Newark Beth Israel Hospital in New Jersey, a 25-year-old woman lay recovering from the loss of her second child. The baby had been stillborn, the body already showing signs of a condition doctors of the time simply called hemolytic disease of the newborn, a slow destruction of the infant's blood that nobody yet understood. The delivery had cost the mother a great deal of blood, and so, following ordinary practice, her doctors gave her a transfusion from the one donor everyone assumed was safest. Her husband. Same blood type. Group O. By every rule, medicine knew in 1937, this should have been simple, safe, unremarkable. It was not. Within moments of receiving her husband's blood, the woman's body turned against it. A severe reaction spread through her, the kind that terrified the physicians in the room, the kind that should not happen between two people whose blood types matched perfectly on paper. A young researcher named Philip Levine, working alongside his colleague Rufus Stetson, began investigating why. They drew her serum and tested it against blood from dozens of other donors, all of them, like her husband, type O. 80 out of 104 samples reacted the same way her husband's had. Something in her blood was rejecting something in almost everyone else's, something the two men could not identify, could not name, could not place inside any blood group system known at the time. They published their findings in 1939. They had no name for what they found, only a careful, correct guess. The woman's body, they proposed, must have built a defense against some antigen she did not carry herself, one inherited by her child from the father, one that had entered her bloodstream during pregnancy and left something behind, an invisible alarm system that reacted violently the next time it encountered that same antigen months later in a hospital bed from a transfusion meant to save her life. It would take another year, and an entirely different set of scientists working with rabbits and the blood of rhesus monkeys before this mysterious factor received the name it still carries today. Rich, short for rhesus. A name born from a laboratory animal that had nothing at all to do with the woman in Newark, or her husband, or the child she lost, and yet forever tied to all three. That is where the story actually begins. Not with ancient bloodlines, not with hidden tribes, not with anything otherworldly. It begins with a nameless woman, a stillborn child, and two doctors trying to understand why blood that should have matched did not. What Levine and Stetson had stumbled onto turned out to be one of the most important discoveries in the history of medicine. Human red blood cells carry a variety of proteins on their surface, and one of them, now called the RhD antigen, is either present or absent, depending on the genes a person inherits. If it is present, a person is Rh positive. If it is absent, missing entirely because of a specific pattern in the genes that build it, a person is Rh negative. Roughly 15 out of every 100 people on Earth carry no RhD antigen at all. The other 85 do. It sounds like a small, quiet detail, one line on a lab report, one plus or minus sign next to a letter. But that small detail turned out to carry enormous weight because of what happens when Rh negative and Rh positive blood meet inside the same body, particularly inside a mother carrying a child. When an Rh negative mother carries an Rh positive baby, something inherited from the father, her immune system can, under the right circumstances, begin to recognize that baby's blood as foreign. Usually the first pregnancy passes without incident because the exposure that triggers the reaction most often happens during delivery itself, when small amounts of the baby's blood mix with the mother's. But her body remembers. If she carries another Rh positive child afterward, her immune system may respond faster and harder the second time, producing antibodies that cross into the baby's bloodstream and begin destroying its red blood cells before it is even born. This is what took the life of the child born to the woman in Newark. Doctors at the time called it erythroblastosis fetalis. Before anyone understood what caused it, before anyone knew how to stop it, this condition killed an estimated 10,000 babies every single year in the United States alone. 10,000 families every year losing children. To a cause hidden so deep inside human blood that nobody could see it coming. For nearly 3 decades after Levine and Stetson's discovery, doctors could only respond to Rh disease once it had already begun, using a treatment called exchange transfusion, slowly replacing a newborn's damaged blood with healthy blood. A procedure refined through trial and heartbreak by physicians who were, quite literally, learning as they went. It worked more often than it failed. Over roughly 20 years, it is estimated to have saved around 200,000 infants, though some were left with lasting damage from the disease itself before treatment could begin. It was not until the late 1960s that medicine finally found a way to prevent Rh disease from happening at all. A treatment called Rh immune globulin, given to Rh negative mothers during and after pregnancy, quietly stops the mother's immune system from ever building the antibodies that caused the harm in the first place. Since it became widely available, the disease that once killed thousands of children a year in America alone has become rare enough that many young doctors today may never see a single case in their entire careers. That is the true, documented story of Rh negative blood. A stillbirth. A transfusion reaction nobody expected. A young doctor determined to understand why. A treatment, decades later quiet and unglamorous, that saved more lives than almost anyone outside of medicine will ever realize. And somewhere along the way, outside the hospitals and the research papers, this same blood type became something else entirely in the public imagination. A legend. A secret. A superpower whispered about across forums and late-night videos, insisting that Rh, negative blood, marks the people who carry it as something other than ordinary, something older, something not quite fully human. The story usually starts with a number. 15% globally carry no RhD antigen, but that number hides enormous variation depending on where you look. In parts of East Asia, Rh negative blood is almost non-existent, found in less than 1 person in every 100. Among many indigenous populations in the Americas and Australia, it is similarly rare. But in parts of Europe, the number climbs dramatically higher, and in one particular population, tucked into the mountains along the border of modern France and Spain, it climbs higher than almost anywhere else on Earth. The genetics behind this trait are, in truth, fairly straightforward, once you understand the shape of them. The RhD antigen is built according to instructions carried on a gene called RhD, sitting on chromosome 1. Most Rh-negative people of European ancestry carry a version of this gene that has been deleted entirely, a small stretch of genetic instructions simply missing from both copies of the chromosome they inherited, one from each parent. Because the trait is recessive, a person needs 2 copies of the deleted version, one from each parent, to actually test Rh-negative themselves. A person with only 1 deleted copy and 1 normal copy will still test Rh-positive, quietly carrying the negative version forward without ever showing it, sometimes for generations, until it happens to pair up with another carrier and produce a child who tests negative for the first time in a family that assumed itself entirely Rh-positive. This is part of why Rh-negative can seem to appear suddenly in a family tree, skipping generations, resurfacing without warning, simply because the underlying genetic instructions were quietly present the entire time, waiting for the right combination. That small, deleted stretch of DNA sounds almost too plain to have inspired decades of speculation. But its unevenness across the globe is genuinely striking, and worth walking through region by region rather than as a single global average. In much of East Asia, including China, Japan, and Korea, Rh-negative frequency drops below 1%. So rare that Rh-negative blood donors there are treated almost like a specialized resource, carefully tracked by name in some regional blood banks because so few exist. Among many indigenous populations across the Americas, and among Aboriginal Australian communities, the frequency is similarly close to 0. Sub-Saharan African populations generally show low to moderate rates, often in the low single digits, to around 4 or 5% depending on the specific region and ethnic group studied. It is really only in populations with substantial European ancestry that the number climbs into double digits at all, generally landing somewhere between 15 and 18% across most of the continent. And then, sitting apart from every other population on that list, are the Basques, whose documented rates climb far beyond anywhere else in Europe. The Basque people have long fascinated geneticists and linguists alike, for reasons that have nothing to do with blood type at first. Their language, Euskara, is what linguists call an isolate, meaning it is not related to any other known language family on the planet, not French, not Spanish, not any of the Indo-European languages that surround it on every side. For centuries, that linguistic mystery led to speculation that the Basque people might represent a very old population, one that had lived in that corner of Europe, since long before Indo-European languages, and the peoples who spoke them arrived. And when researchers began studying Basque blood in the 20th century, they found something that seemed to fit neatly into that same old mystery. Basque communities carry some of the highest documented frequencies of Rh-negative blood of any population on Earth, commonly cited at around 30 to 35%, more than double the global average. Put those two facts side by side, an ancient, unrelated language and an unusually high rate of a rare blood type, and it is easy to see how the story took on a life of its own. Online communities, largely gathering on forums and blogs dedicated specifically to this subject, built an entire mythology around it over the past 2 decades. Rh-negative blood, some claimed, was not simply rare. It was proof of a hidden bloodline, one that stretched back before recorded history, one that some believed connected to the Basques as the last surviving remnant of Europe's original, pre-human, or even non-human inhabitants. From there, the claims only grew stranger. People wrote that Rh-negative individuals ran cooler body temperatures than everyone else, that they had an extra rib, or an extra vertebra, hidden proof of a different origin, that their blood carried a faint blue tint, that they experienced heightened senses, unusual sensitivity to heat and sunlight, vivid dreams, even psychic ability. Some tied the bloodline to ancient stories of the Nephilim, a term drawn from a handful of verses in the book of Genesis describing mysterious offspring born of unions between divine beings and human women, figures the text describes only briefly and without much detail, leaving enormous room for later writers and internet communities to fill in a backstory the original text never actually provided. Others skipped mythology altogether and went straight to extraterrestrial ancestry, suggesting Rh negative blood was the genetic fingerprint left behind by beings that were never human at all. If Rh negative blood really was ancient, really was tied to a population older than the rest of modern Europe, could it have come from somewhere unexpected? Could it have come from the Neanderthals? The idea is not hard to understand, even if it turns out to be wrong. Basque populations, along with several other groups, have been shown in genetic studies to carry somewhat elevated levels of Neanderthal-derived DNA compared with some other populations. A legacy of interbreeding that occurred tens of thousands of years ago when early modern humans expanded out of Africa and encountered Neanderthal groups already living across Europe and parts of Asia. If Basques carry unusually high Neanderthal ancestry and Basques carry unusually high rates of Rh negative blood, it is a short, tempting step to assume one caused the other. For years, that assumption spread widely online, repeated so often it began to feel like established fact rather than an unproven guess. Then scientists actually went and checked. In 2021, a research team working across institutions in France, including the CNRS and Aix-Marseille University, published a study in the journal PLOS ONE that did something nobody had done before with this specific question. Rather than guessing at Neanderthal blood type from indirect evidence, they directly reconstructed the blood group genetics of 3 Neanderthal individuals and 1 Denisovan, an extinct human relative discovered in Siberia, using ancient DNA extracted from their remains. The team included a paleoanthropologist working alongside population geneticists and specialists in hematology, the study of blood, combining fields that rarely overlap in order to answer a question that had sat unresolved for years. Piecing together a reliable blood type from DNA that is tens of thousands of years old, fragmented, and often contaminated by the environment it was buried in, is painstaking work, closer to careful reconstruction than a simple readout, and it took specialized techniques to confirm the results were reliable enough to publish. What they found did not support the popular theory. It quietly, thoroughly contradicted it. The Neanderthal and Denisovan individuals studied carried a distinct Rh gene variant, one that is essentially absent from modern human populations today, found so far in only a small handful of individuals, including 1 Aboriginal Australian person and 1 person from Papua New Guinea. According to the same body of research, the Rh gene variants that are common in modern humans today most likely arose within early Homo sapiens, probably not long after our ancestors first left Africa, possibly while passing through the region of the Persian Plateau, long before those populations ever reached Europe or encountered Neanderthals at all. An earlier line of inquiry reached a similar dead end from a different direction. Years before the 2021 study was published, a genetics enthusiast reached out directly to Svante Pääbo, the Swedish geneticist whose work sequencing the Neanderthal genome later earned him the Nobel Prize, asking a simple, direct question. Were the Neanderthal specimens his team had sequenced Rh negative or Rh positive? The answer came back clear. Based on the genetic marker used to determine Rh type in modern humans, both the Denisovan individual and the Altai Neanderthal specimen his team had studied appeared to be Rh positive, not negative. 2 separate lines of research, using 2 different methods, years apart, both arrived at the same quiet, almost anticlimactic conclusion. If anything, the evidence points the opposite direction from the popular legend. Neanderthals, as far as science has been able to determine so far, were more likely to carry Rh positive blood, not the rare negative type so many people online have spent years trying to trace back to them. Geneticists who specialize in this exact question have said as much plainly, without much drama, in the way careful scientists usually do. Rh negative blood almost certainly did not come from Neanderthals, and it almost certainly did not come from anything outside the human family. It is simply one more example of ordinary genetic variation, the same basic category of difference that gives some people red hair or blue eyes, or the ability to taste certain bitter compounds that others cannot taste at all. Some researchers estimate the mutation responsible for the common European form of Rh negative blood may be far older than the Neanderthal encounters the myths point to, potentially reaching back millions of years rather than tens of thousands, meaning it likely existed in some form long before modern humans as a species had even fully emerged. So if it was never Neanderthals, and there is no credible evidence it was ever anything from beyond this planet, what actually explains the Basque mystery? Why does Rh negative blood cluster so heavily in that one corner of Europe, and so lightly almost everywhere else? The honest answer is less dramatic than any ancient bloodline, but it is not without its own quiet wonder. Population geneticists have a specific name for what likely happened in a place like the Basque Country, a process called a founder effect. Imagine a relatively small population, isolated for generation after generation by mountains on one side and a very different language and culture on every other side, marrying mostly within their own communities rather than blending steadily with surrounding groups. Whatever genetic variations happen to be present among the founding families of that population, whether common or rare, tend to stay unusually common within it, simply because there are fewer outside genes flowing in to dilute them over time. If the ancestors of the Basque people happen to carry the deleted RHD gene at a higher rate than their neighbors, purely by the ordinary chance of who founded that population and who they had children with, that higher rate would tend to persist for thousands of years afterward, largely undisturbed, long after the original reason for it had been forgotten. It does not require ancient astronauts, or a lost tribe from before humanity, to explain why a rare genetic trait might survive at unusually high frequency in a population that stayed distinct while the world changed around it. It only requires time, geography, and the ordinary patient mathematics of inheritance, quietly repeating itself across hundreds of generations, long before anyone was there to notice or explain it. It is worth sitting with why the other version of this story, the superpower version, spread so far and took hold so deeply in the first place. Beyond the extra rib, the psychic sensitivity, and the alien ancestry, some corners of this mythology added still more specific claims over the years that Rh-negative individuals run measurably cooler body temperatures, that their eyes are unusually sensitive to bright light, that they show higher rates of certain rare allergies, that their blood carries a faint blue tinge instead of the deep red of ordinary hemoglobin. None of these claims have held up under any kind of controlled scientific study. Blood appears blue only briefly when deoxygenated, inside veins, beneath the skin, an optical effect of how light passes through tissue, and it happens in every person regardless of Rh type. Body temperature, light sensitivity, and allergy rates vary for reasons entirely unrelated to a single antigen on the surface of a red blood cell. Even the modern rise of consumer DNA testing kits, the kind people order online and mail back with a saliva sample, has done little to settle the mythology, mostly because those tests were never designed to investigate it in the first place. They report ancestry percentages built from comparison databases, not verdicts on hidden bloodlines, and results showing high Basque or high Neanderthal ancestry says nothing at all about whether a person's Rh type came from some exotic source rather than the same ordinary inheritance everyone else's blood type comes from. A blood test that comes back negative, a small technical detail on a medical form, feels like very little on its own. But turn that same fact into a secret bloodline, a whisper from before recorded history, and suddenly an ordinary trait becomes something people want to claim as their own identity. It is not so different from wanting to believe an old family story about a distant, mysterious ancestor. The desire for a hidden, special origin is very old and very human, and it does not need scientific evidence to keep spreading once it starts to feel meaningful to the people carrying it forward. But perhaps the real story does not need the embellishment at all, because somewhere in New Jersey, in the autumn of 1937, a young mother whose name has mostly been lost to history lay in a hospital bed, grieving a child she had just lost, her own body reacting violently to blood that should have saved her. She could not have known that her reaction, confusing and frightening as it was to everyone in that room, would eventually be traced back through decades of careful research to a single missing protein on the surface of her red blood cells. She could not have known that understanding her case would eventually spare hundreds of thousands of other mothers the same grief she carried out of that hospital. She was not a legend. She was not descended from anything beyond this world. She was simply a person at a specific moment in medical history whose body asked a question that took science 30 years to fully answer. That, in the end, is what Rh-negative blood actually is. Not a mark of hidden power, not a bloodline reaching back before history, not a fragment of something not quite human hiding in plain sight inside 15 out of every 100 people alive today. It is a small, quiet variation, inherited the same way eye color or height is inherited, carried without any drama at all by thousands of generations of entirely ordinary people, most of whom never gave it a second thought, right up until the moment it mattered enough, in a hospital room in 1937, for two doctors to finally start asking why. There's a strange comfort in stories like this one, if you let yourself sit with them long enough. Not every mystery needs to end in something vast or otherworldly to still feel meaningful. A missing protein on the surface of a cell, invisible to the naked eye, invisible even to the microscopes of 1937, was enough on its own to end a young mother's second pregnancy, to puzzle two careful researchers for years, to eventually be traced through the blood of Neanderthals buried in caves for tens of thousands of years, and to finally be understood well enough, decades later, to spare countless other families the same grief. That is not a small story simply because it lacks aliens or ancient astronauts. If anything, it is larger, because every part of it actually happened, documented in journals, preserved in hospital records, verified again and again by scientists, with no reason to exaggerate any of it. Somewhere tonight, a small percentage of people reading or listening to this will check quietly, almost without meaning to, which type their own blood is, positive or negative, ordinary, either way. Carried forward from parents who carried it from their own parents, back through generations none of us will ever meet, each one utterly unaware they were passing along a detail that would one day matter enough to save lives, and strange enough, later, to spark 100 years of stories far larger than the truth ever needed to be. If this is the kind of quiet mystery you like drifting off to, let it sit with you for a while before you close your eyes tonight. And if you would like more calm, carefully researched stories like this one to fall asleep to, consider subscribing so the next one finds you when you need it.